Let's start with viruses that have double-stranded DNA genomes. And I want to mention 5 different viruses in this category. The first are the adenoviruses. Now we're using here the family designation for these viruses. So adenoviruses would be Adenoviridae. Families end in a viridae. And Adenoviridae are unusual looking viruses, as you can see. They cause human respiratory and gastrointestinal tract infections. Then we have the herpes viridae. These herpes viruses are in all of us. You have a herpes virus infection. You probably have many, and I certainly do. We acquire these when we're young, and they remain with us for our lifetimes. The papillomaviruses are rather small viruses. These are the agents of warts, and importantly, cervical and anogenital cancers. The polyomaviridae contains viruses that infect everyone, almost all of the human population, but they only cause disease in people who are immunosuppressed. And finally, the poxviridae family contains the important human pathogen smallpox virus, which was eradicated in the 1970s. It no longer exists on the planet Earth except in 2 laboratories. So these viruses all have in common the fact that their genome is double-stranded DNA. And you can see that on the right-hand part of the screen here. That DNA, when it gets into a cell, directs the production of messenger RNA. We can make messenger RNA from double-stranded DNA templates only. The mRNA, of course, is translated by the cell's protein synthesis machinery to make proteins, and those proteins go to making new virus particles. Along with this, of course, the DNA genome has to be replicated. So we see here we're going from one double-stranded DNA to another. That's called DNA synthesis or DNA replication. And the newly made DNAs are then packaged into the virus particles. So that's the overall replication screen. Now, among the viruses that have a double-stranded DNA genome, there are ones with rather small genomes. For example, the polyomaviruses and the papillomaviruses have genomes that are 5 and 8,000 base pairs in length. This is really too small to encode a lot of proteins, and one of the important ones that's missing is the enzyme that replicates the DNA genome. It's called DNA polymerase. These viruses have to employ the DNA polymerase of the host cell. So their genomes are copied by host cell DNA polymerases. On the other hand, there are other larger viruses in this category, including the adenoviruses. Their genome is a double-stranded linear DNA molecule from 36 to 48 kilobase pairs in length. The herpes viruses have substantially larger DNA genomes from 120 to 220 kilobase pairs. And then even larger are the pox viruses, between 130 and 375 kilobase pairs. The poxvirus DNA is unusual. It is also a linear double-stranded molecule, but the ends are covalently connected. And so if you separated the 2 strands of this virus DNA, it would form a single-stranded circle. All of these virus genomes are very large, and they encode a viral DNA polymerase. So in terms of that function of DNA replication, they're independent of the host cell.